Good morning, good afternoon, and good evening, everyone, and welcome to another edition of JAX Live. I'm your host, Natalie Herlin from the Jackson Laboratory, and joining me today is Dr. Grace Berryhill of Technical Information Sciences. Good morning, Grace. And Dr. Andrew Schile, Senior Scientific Advisor. Welcome, Andy.
Hey, Natalie.
Thank you both for being here. So in today's live stream, we're going to be discussing immunodeficient mice. And these mice are valuable research tools in a variety of fields: immunology, infectious disease, oncology, and more. And they can also serve as hosts for engraftment of different tumors. You can engraft mouse tumors, human cancer cell lines, patient-derived xenograft tumors or PDX, and even components of the human immune system. So with all of these factors, this is leading us to the question of, with a lot of models to choose from, how do you pick the best one for your study? And that's what we're going to be discussing here today in our live stream. I also want to point out that in the video description below, you can see a link to a recent blog post we've done on this topic that goes into a little bit more detail about the factors involved in choosing an immunodeficient mouse model. Audience, a reminder, please be sure to use that comment box. You can submit a question, you can add a comment, and we'd love to hear where you're joining us from today. So give us a shout out from wherever you are in the world. Thank you all for being here. We're going to try and answer as many questions as we can live during today's discussion. All right, gang, let's go ahead and get started. So I feel like the right place to start is to talk about these immunodeficient mouse models and what types of immunodeficient mouse models available. I think many of us may have heard of nude mice. Are there more than that? What are some of the categories? Grace, would you mind leading us off?
Absolutely, absolutely. It's a really critical question. There are many immunodeficient mouse models available. You're right. Most of us are familiar, at least colloquially, with nude mice as a broad category. And yes, they have been a key player in the field for a very long time now, but that just represents one strain of really a whole repertoire that exists. So nude mice are characterized by a defect in T cells, but there are other mutants that exist. So PRKDC SCID mutants that are deficient in functionally mature T and B cells. RAG deficient mice, RAG1 and RAG2, also a T and B cell deficiency. But there are many other higher order multigenic immunodeficient mice as well, things like NSG mice, for example. So there really is a broad repertoire that exists, all with different features, different characteristics that are crucial to understand as you're looking to make a model selection decision, conceptualize an experiment, understand what options are available for you. So I'm hopeful that some of what we can offer today will help guide those conversations.
Well, it's great to have access to a wide variety, but of course that's the same thing as being at the supermarket and trying to pick something in an aisle that's full of different products, right? You need to know the different products and make that selection decision. And it sounds like one of the first things you could consider is genetic background. So Andy, maybe you can talk to us a little bit more of why genetic background matters when it comes to picking an immunodeficient mouse model.
Yeah, I'd be happy to talk about that. So Grace introduced us to this idea that there are certain key mutations that create immunodeficiency in mice, like the nude mutation, the SCID, the RAG mutations, et cetera. But the important thing to remember is that, even when these genes are mutated in mice have 20,000 plus other genes as well. Mm-hmm. And so the combinations of alleles that naturally occur in different strain backgrounds, for example, are going to be different between black 6 and BALB and NOD and other strain backgrounds. So those naturally occurring alleles can, in some cases, modify or affect those primary drivers of immunodeficiency, like those SCID mutations. And in some cases, like in the NOD background, they can introduce immunodeficiencies of their own. So this NOD, or non-obese diabetic strain background, has naturally occurring alleles that contribute to deficiencies in mouse macrophage function, dendritic cell function. There's an absence of complement system. There's reduced activity of mouse natural killer cells. And in some cases, these strain backgrounds can contribute alleles that are completely unrelated to immunodeficiency, but still help to make them ideal platforms for certain experimental models like the NOD background, has a naturally occurring variant of a SERP alpha protein that helps to make the bone marrow niche for human hematopoietic stem cells and graft in humanized models, helps to make that niche more welcoming for human cells compared to other strain backgrounds. So in some cases, the strain background can be quite important, and we have resources on our website to help you understand what are the differences between, say, for example, a SCID mutation with the BAL-C genetic background versus that same mutation that might be present on the BLAC6 background or the NOD.
I'm nodding right along with you here, Andy, as you discuss all these different genetic variations that are available. And thank you for referring our audience to the website for more information, because it's one thing to be aware of all of these varieties, but it's another thing to start going through and looking at all the choices and making the right selection. So Grace alluded to the fact that when it comes to nude mice, there's more than one type available. There's an entire repertoire of nude mice. So these all seem to have some similar nomenclature. There's the NUJ inbred mice, there's B6 nude congenic mice, and even JNU outbred nude mice. What are some of the differences between these strains that seem to have pretty similar names?
Yeah, I'm happy to answer that question also. So there are 3 primary varieties of nude mice that we distribute at the Jackson Laboratory, like Natalie alluded to. So the majority of nude mice that are in use in the field are principally for cancer research, and those have an outbred genetic background. So outbred, of course, means that the individual mice are going to be genetically different from each other in ways apart from that nude mutation that they carry, right? So there's going to be more genetic variation in a population of outbred nude mice. And so outbred stocks in general tend to be more robust than inbred strains. And that's simply because there is more genetic diversity. So an outbred nude, like our outbred JNU, is very commonly used in the field. And this is a generally all-around robust strain due to the outbred genetic background, with genetic heterogeneity among mice in a group. And so we also have an inbred nude mouse that was created by inbreeding nude mice from an outbred population. So an inbred strain population, all those mice are essentially going to be genetically identical to each other. And so, like I mentioned, generally speaking, outbred stocks are more robust than inbred strains. So, for example, they might tolerate harsh chemotherapy treatment, for example, to a greater degree than an inbred might. But it just so happens that the inbred nude mouse that we distributed at the Jackson Laboratory, the NUJ, happens to be a very robust animal itself in terms of its body weight and general ability to withstand cancer treatment in the most typical application for this strain, which would be for growing human cancer cells in the mouse. The third option that you might see on our website is the C57 BLAC6 congenic variant. So it has the same mutation as that outbred nude and the inbred nude, but the difference is that the C57 BLAC6 genetic background. So a human cancer cell will very likely grow very well in any strain background. If it's an established cancer cell line that you know grows well in a nude mouse, it'll probably grow in any other nude mouse. There could be some differences, of course, that you could verify experimentally, but the major application for the BLAC6 nude mouse would probably be in some situation where that strain background is particularly important. For example, it could be something related to the hairless phenotype that might be important, or the deficiency in the thymus, which of course is where the T cells develop. Or for example, if you're crossing it to another C57 BLAC6 strain that may have a different mutation that you want to combine with the nude mutation. But generally speaking, the C57 BLAC6 nude strain is not one that is most commonly used in the field for cancer xenograft studies. That's not to say it can't be used, but that's a strain that you might consider if there's something about that BLAC6 genetic background that's important. Again, it may not matter in many situations, but it's one of the benefits and also the challenges of having a lot of strains to choose from, is that you need to understand what are the benefits and limitations of those different strains. And fortunately, we have resources to help with that.
And thank you for that really great overview, especially explaining the differences between the outbred, the inbred, and the congenic strains. That was really helpful to learn between them. We're getting some shout outs. We have some viewers today in Minnesota, India, Austria, Italy, Maine, and California. So thank you, everyone. Thanks for joining us here. You've got quite a few time zones represented. So we've talked a lot about genetics and genetic background and alleles and how that can impact your selection of a model. I'm wondering, big picture, Grace, what else do we need to consider? What advice do you have for someone who's starting this process of selecting a model for their research?
Sure. It's a question that the team that I work on in technical information services helps people navigate frequently. People will come to us and say, what's the best host for me to use, to engraft tissue X, Y, or Z?
Yeah, it depends, right?
And the answer in virtually all cases is it depends.
Mm-hmm.
And it depends on a variety of factors. I would say from a big picture perspective, when we're speaking about using immunodeficient strains, we're frequently discussing them as a xenograft host, right? So introduction of non-mouse tissues or cells into the mouse. And so I would say it's crucial to understand the characteristics of the tissue that you'd like to introduce or the cells that you'd like to introduce. So as Andy mentioned in discussing, different variations of nude mice, if you have, an immortalized tumor cell line that's happy to grow in cell culture, happy to grow on plastic, that's pretty robust. And so, that's an important feature to know when you're saying, okay, so maybe I can choose some of these lesser immunodeficient models. That gives me an understanding of where I can start here. But then if you're looking at introducing, maybe a primary human tissue, so tissue that hasn't been put in culture, for example, then you're probably going to want to avoid some specific strains. So like nude mice are probably not going to work exceptionally well in that application. Then you'd be looking for things that are at a higher level of immunodeficiency, to most supremely immunodeficient like NSG mice. Mm-hmm. And then of course, on top of that, I would say it's critical to really look at the readout that you'd like to measure in your experiment. What are the cells that you need to be present? The cells that need to be of mouse origin, the cells that need to be of human origin, and what can we do to essentially create or find a host that best supports kind of the intertwining of all of those aspects. So it isn't a straightforward answer. And it's tough, but the good thing is there are a lot of options available. And so our team is always happy to try to help, bring people to those varieties of options.
And that team is Technical Information Sciences. And for those viewers in our audience, if you want to reach out to Grace and the rest of the TIS team, you can contact them at micetech@jax.org for questions and guidance and assistance in doing model selection. So what I heard from you, Grace, is there's a lot of working backwards from your research goals to kind of eliminate some set of models. And then you can start to get into all of these details of what's left. How do I pick the one that's for me? And we've talked about genetic backgrounds. Another factor that could be important is lifespan. And that's going to vary depending on the model you select. And I'm wondering if the two of you can walk me through how you'd factor lifespan into your model selection.
Yeah, I'm happy to start answering that question, Natalie. So different strains, and this is true for any aspect of mouse biology, different strains are going to have different characteristics. And one of those characteristics, of course, is lifespan. In some cases, for example, SCID mice, so mice that have a deficiency in T and B cells, on certain strain backgrounds like that NOD or non-obese diabetic background, the NOD SCID, as these mice get older, they have a tendency to develop cancers of their own that they're going to be developing with some frequency, regardless of whether or not you've engrafted any human cancer cells or any human cells at all into this mouse. And so it's important to know in some of these models, for example, with a median onset of about 9 months, these NOD SCID mice are going to be developing tumors of their own. So this doesn't have to be a limitation in some circumstances. For example, if you know that you can execute your cancer study in a non-skid mouse because you've chosen this as the most appropriate one for your project, if you know you can execute that in a matter of several months, you probably won't run into some of these limitations, if you want to call them, of the model. But in some cases, for example, if you're working with principally thinking about tumor models, but if you're working with a potentially slow-growing tumor, so for example, one that might come from a clinical sample, you might want to combine the severe immunodeficiency of one of the newer generation of models, like the NSG, the non-skid gamma mouse, because not only is that mouse have a severe reduction in its own mouse immune system, but it's a long-lived mouse. Mm-hmm. So for example, if you have a very slow-going tumor model, it can benefit from a strain like this, which doesn't develop those thymic lymphomas at the same frequency that, for example, a non-skid mouse might. So I think it would depend on the selection of the model, of course, depends on how long you think the experiment will last. And so that's just another thing to consider when thinking about these different models. So I know from, for example, we have this program at the Jackson Laboratory, the patient-derived xenograft program, mm-hmm. model, that has received samples from the operating room of resected tumors or biopsies, and used that NSG mouse to establish patient-derived xenografts. In some cases, there have been some very slow-growing tumor models. Yes. That are just related to the biology of that particular patient cancer. And in those cases, they benefited from having a strain where we had the expectation that this mouse would be a long-lived mouse that wouldn't be developing too many of these kind of background pathologies that would just be occurring whether or not there were tumors engrafted in it.
Yeah, thank you for that example. So it sounds like you need to know how long your experiment will last, but it's also important to keep in mind that the model itself can change over time, right? So there's so many layers to these discussions and considerations. I think we have time for one more quick question here before we wrap up today. Another topic that I've heard mentioned is the concept of leakiness. And I'm wondering if you could briefly tell us, what does leakiness mean in the context of immunodeficient mouse models, and why are some models more susceptible than others? I think it ties into what you were getting at there, Andy, a few minutes
ago. Yeah, I'm happy to follow up on that. So this is just another example that the immunodeficiency phenotype can vary depending on the strain background of the mouse that you're working with. So leakiness is the tendency of any mouse with the SCID mutation to develop mature T and B cells as it ages. Okay. So this is typically reflected in, for example, if you have mouse B cells developing, you can see evidence of mouse immunoglobulin in the serum as these animals get older. And then, for example, there are some strain backgrounds like that NOD SCID mouse where leakiness is quite low, virtually negligible. So this would include either the NOD SCID or the NSG, which is the NOD SCID gamma mouse. He's going to have a very low tendency or virtually negligible tendency for these mouse T and B cells to develop as they age. But the same mutation, same SCID mutation on, for example, a BALB/C genetic background or BLAC6, there is this tendency for mouse B and T cells to mature as they get older. And so the impact on this is not entirely known in a lot of cases. It may not be a major limitation for the BLAC6 and the BALB/SCID models for short-term experiments, but it is something to keep in mind. This is just one of many examples in mouse genetics that strain phenotype can and will, in some cases, depend on the strain background that you're studying.
Thank you. Well, we've covered a lot of ground here today, and I know that this is just the tip of the iceberg when it comes to all the considerations that you have to keep in mind when selecting a model. We've mentioned technical information scientists as a resource for our audience members who want to learn more about the different models and might need some guidance and selection. So I want to reiterate that you're not alone. If you're going through this process, reach out to us and our team of experts for help. Grace is available to help you, guide you through this process. And we've also included, as I mentioned, a blog post on this subject in the description of the video. That's another resource for you to read in a little bit more detail some of the topics that we've covered here today. Well, unfortunately, we are out of time. I want to thank you and you, Grace and Andy, for joining us for the productive discussion. Thank you for sharing your expertise with us. And to our audience, thanks for tuning in. And I wish you a good morning, a good evening, and a good night. We'll see you in another edition of JAX Live soon. Thanks.
Thank you.
Thanks all.